We'll switch over now to the topic of antibiotics. This topic is related to prokaryotic cell biology because antibiotics are going to be medicines that kill or disrupt the growth of bacteria. These medicines are going to specifically target the structures or the functions that are unique to prokaryotic cell metabolism. They target bacterial cells, but they do not harm the host cells. So an antibacterial, something like rubbing alcohol or ethyl alcohol that we find in the different types of hand sanitizing lotions, those antibacterials are generally effective against all microbes, and generally they are poisonous to all living things. So if you have a bacterial infection, an antibacterial is not going to be helpful because that may cause damage to many other types of cells within the body. But rather, an antibiotic will target specifically bacteria. And the reason why these antibiotics exist is because they are mostly naturally occurring. They are derivatives from fungus because fungi and bacteria are competitors. Both fungi and bacteria, they are both saprotrophs, and they both are found in similar ecological niches. So because of that, they compete against each other, and there's a type of chemical warfare that happens as fungi and bacteria each produce classes of chemicals that are meant to be able to poison each other. Because if a fungus can poison the bacteria, then that means that there will be more food left for the bacteria and more food left for the fungus and less competition for that food source. So there are many different classes of antibiotics. The type that I will focus on are beta-lactams, which are going to be the most widely used antibiotic in the country and across history. This includes such drugs as penicillin, ampicillin, and other derivatives of it. Each of these different classes is going to do something that either inhibits the growth of bacteria so that the infection doesn't spread, or outright causes the death of that particular cell. So you can see that there are some that restrict growth and reproduction, which are the lighter green, and the darker green are those that are going to cause bacterial death. Each of these different groups is going to target a different part of the metabolic function of the bacteria. So each of those different modes of actions will help it to be particularly effective against a subgroup of bacteria. The discovery of these has extended across the last 100 years, and so we've seen that our toolbox, so to speak, of antibiotics has increased over time as our study of fungus and our chemical synthesis of a few of these has increased over time. This diagram shows you the breakdown of some of those classes of drugs based upon the location in which they target bacteria. The beta-lactams are going to work against the synthesis of peptidoglycan in the cell wall, and I'll return to that in a few minutes. Other aspects of cell biology that are unique to bacterial cells include protein synthesis. So there can be drugs that target the ribosomes that are unique to bacterial cells since they have a different type of ribosome. Additionally, there's a different type of way of building DNA. So the process of DNA replication is different. The metabolic pathways are different, so different types of enzymes producing different types of important nutrients. And so every area in which the bacterial cell is unique from a human cell is a potential area where an antibiotic can be utilized to be able to target and cause the death of those cells. So we'll look now in the last few slides here on penicillin. Penicillin is notable because it was the first known discovery of isolation of an antibiotic. There are three important people in this. All three of these together won the 1945 Nobel Prize in Medicine. It starts with Alexander Fleming, who is known for his discovery of the antibacterial properties of penicillin, and then Florey and Chain, who worked together to be able to purify and synthesize penicillin on a large scale. So we'll start with Fleming. And Fleming benefits from a topic that comes up from time to time across scientific history, and that's serendipity. So Fleming, as the story goes, was studying bacteria, and so he would make petri dishes that would contain different types of bacterial strains that he needed for his experiments. Now, it is common, particularly when you don't utilize the most hygienic of methods, that those bacterial plates can get contaminated by mold spores. Those mold spores are floating through the air all of the time, and so if those mold spores land on the surface of the petri dish, they are just as likely to grow as their bacterial counterparts. Now, what Fleming did is that he noticed, before he discarded his plates, that a particular fungus that grew caused a zone around it to be free of bacteria. And as he repeated this, he saw that this particular type of mold from a genus of molds called Penicillium, which we find oftentimes on many forms of food, like in the picture the orange here, what he found is that continuously this Penicillium mold would prevent the growth of the bacteria which he was growing in his dishes. This was followed up by Florey-Chain, who in the 1940s were working to try to be able to capitalize on those properties, to be able to actually produce a medicine that could be useful to people. They took those molds and isolated a little bit of a particular drug, which we called penicillin. And that penicillin they then injected into mice, which had been given a particularly virulent strain of Streptococcus bacteria. If no penicillin was given, all of those mice died. But when they gave the penicillin injections to the mice, no matter the quantity of the drug given, the mice survived with no ill effects. So this was an important part of being able to develop the drug and to ensure that it was safe. First, that it caused no harm to the mice, and secondly, that it was effective in being able to kill the bacteria that otherwise would have led to the death of the mice. The successful production of penicillin coincided with World War II, and so there were great efforts made in the early 1940s, so that by 1944, during the invasion of Normandy, there were large quantities of penicillin taken with the soldiers during that invasion, because death was just as, if not more likely, going to happen due to infections of wounds rather than death on the battlefield itself. So being able to provide this medication that was capable of being able to save lives from those bacterial infections, this is one of the crowning achievements of the 20th century and has led to the saving of many millions of lives in the decades since. We can take a quick look now at the mechanism by which penicillin works. So penicillin is going to be a drug that acts upon the cell wall of bacteria. Bacteria have a cell wall that's made of a substance called peptidoglycan. Peptidoglycan is similar in some ways to cellulose. It's a long, linear chain of carbohydrates. But the strength of peptidoglycan comes in the fact that there are short chains of amino acids that link together the chains of those polysaccharides. And there is a protein, an enzyme, that is responsible for building those little chains that are going to link together the polysaccharides. What penicillin does is that penicillin inhibits the enzyme that's necessary to build those chains. So when penicillin is introduced to the bacteria and the bacteria attempts to grow, it cannot synthesize the cell wall. And because the cell wall cannot be synthesized, that creates a weakness. And that weakness will make the bacterial cell susceptible to osmotic pressure. And so in a very short period of time, any change in the salt balance inside or outside of the cell is going to lead to either the dehydration or the bursting of those bacterial cells. So therefore, disrupting the cell wall directly then leads to the death of that cell and the inability of that cell to successfully reproduce and make more bacteria. So by introducing this drug to somebody with an infection, ensures that the bacteria will die, and the immune system will be able to ensure that the remaining bacteria are targeted and destroyed before that infection can spread and cause further pain and death. Now, one of the issues is that penicillin is really no longer effective, and it is no longer commonly prescribed as a medication for things like infections. So a streptococcus infection, if you have strep throat, or if you have an ear infection, there is not going to be penicillin which will be prescribed for that. And that's because antibiotic resistance comes about in a fairly short period of time in bacteria. It comes about due to natural selection. So in an organism, so say in a human body, there might be a lot of streptococcus bacteria causing strep throat. The majority of those bacteria, represented by the gray shapes here, are going to be bacteria that are going to be susceptible to the antibiotic. However, there may be a few bacteria that contain a particular mutation which changes the shape of some molecule. For example, in this case, changing the shape of the protein that builds those cell walls. And therefore, when the antibiotic is introduced, all of those who are susceptible die. But not all of the bacteria die, because those that have that unique genetic code, they are going to survive. And when they survive, they are going to reproduce. And so they become more and more common from generation to generation within bacteria. But not only that, because of the process of conjugation, any bacteria that have that resistance to the antibiotic are capable of sharing that through a plasmid to other bacteria, thus further increasing the concentration of antibiotic-resistant bacteria in that population. So if this was a human body, then at this point, then that first antibiotic, which was ineffective, would have to be replaced by a different antibiotic, which is targeting a different aspect of prokaryotic cell metabolism. And so that is your introduction into prokaryotic cells and how to treat prokaryotic cell infections. As always, if you have any questions, please let me know. Thank you for watching.